Now we are going to study about the role of lymphocytes. Already we have studied T cells and B cells. Now we are going to study that only, how they are going to work out. Now B cells and T cells, little explanation we have to study about it. Now this is the diagrammatic representation of a part of the bone. Okay, very big bone we are taking. We are taking the section. A wide cavity is present here. Now this is the cavity. Now here and all hard bone is present. Bone is very hard. But here the cavity is there. The cavity is filled with a soft tissue, very soft tissue. That is called marrow. In the marrow, many types of cells are there. But I am going to take only one lymphocyte here. Now this is the lymphocyte which is just produced. It is an immature lymphocyte. It is there in the bone marrow. But in the bone marrow itself, it is growing little. Okay, it is growing. But again it is growing and reaching the maximum size. It is reached the maximum size. Now once it is reaching the maximum size in the bone marrow itself, it is called a matured B cell. Why we have to call it as B cell? This cell is matured in the bone marrow. So matured B cell. It is also called a matured B lymphocyte. Once the cell is matured, the bone marrow is sending the cell to the blood. So that it will come to the blood. It is circulating along with the blood. Whenever microorganism enters, this matured B cell will fight with them and kill them. This is one type, how B cells are produced. Now in the bone marrow, again I am taking one small lymphocyte is there. It is immature lymphocyte, but it is not at all growing in the marrow. Like this, it is not all growing, it is young. Then what the bone marrow will do, it will push the young or immature lymphocyte to the blood. It will mesh, that is push it. Now the young lymphocyte, immature lymphocyte is at the blood, but it cannot act on the microorganism because it is immature, young. Then the blood is depositing this young lymphocyte into the cortex of the thymus gland. Now the thymus gland, already we have studied the structure. Now here the thymus gland, it has come. Now in the thymus gland, it is growing little by little and attaining maturity, full size in the medulla. Now it is there in the medulla. Now once it is matured, it is called matured T cell. We are calling it as a matured T cell because it is matured in the thymus. So that we are calling it as matured T cell or matured T lymphocyte. Once it is matured, the thymus gland is pushing the matured cell into the blood. So B cells are coming to blood, T cells are also coming to blood. B cells and T cells join together, they can attack the microorganism. So that is the explanation for B cells, how they are formed, T cells, how they are formed. Now we are coming to activation of B cells, how the B cells are working. For immunity is concerned, how the B cells are working, we have to see. Now this is the B cell, okay, now this is the B cell, B lymphocyte. Okay. Now what the B lymphocyte is, on the outer surface it is having one small region. It is called acceptor protein. It is a special protein present. Don't think that only one place it will be there, but many places it will be there. So special protein is scattered at the outer surface of the B cell. It is called acceptor protein. Now the acceptor protein is always touching the pathogen. Once a pathogen is entering, the pathogen is attracted by the acceptor protein. So the pathogen, it can be bacterial cell, or it can be virus, or it can be protozoan parasite, or it can be fungal parasite, anything. It is microorganism. It is foreign body. It is attracted and it will come and attach with the acceptor protein. Once the pathogen is attached with the acceptor protein, then the B cell is activated. So far the B cell is common, normal, inactive, not doing any work. But once the microorganism is sticking with the acceptor protein, now the B cell is activated. Then the B cell is keep on multiplying. Okay, B cells are keep on multiplying again and again. Enormous number of cells are produced. Now the resulted cells are called plasma cells. The resulted cells are called plasma cells. So B cells are changed into plasma cells. Now the plasma cells are synthesizing. Synthesizing means secreting, producing antibody. Okay, it is producing one antibody. This antibody will attack the pathogen. Once the antibodies are produced sufficiently, it will come and attack the pathogen and it will destroy the pathogen. That is a basic mechanism. Now this antibody, what is secreted here is a specific antibody. For example, here the pathogen what has come and attached here is tuberculosis bacteria, bacterium which is causing TB, tuberculosis. Then all the process will take place. This is the antibody. The antibody is specifically killing only the tuberculosis causing bacteria. That is only specific. It won't kill any other type of bacteria. That is called a specific antibody production. That is the role of B cells. Now that is B cells. Note, some B cells are not activated. Okay. Now even if microorganisms are coming and touching, they are not at all activated, but they remain as memory cells. But what they can do is, even if the microorganism is touching, that is, it is not activated, it is not multiplying, it is not producing antibody. But what will happen? It is understanding the chemical composition of the pathogen and keeping the chemical composition in memory. So they are called the memory cells for second immune response. What is that second immune response? We have to see. Now, first time the immunities are developed, antibodies are produced, but antibody is not sufficiently produced. But the microorganism is destroyed. Maybe after a few years, once again the same pathogen is entering. What will happen? These memory cells which are present here, they will also join and they will also produce the antibody second time. When the second infection is taking place, that is called a second immune response. So first immune response they are not acting, but the second time infection takes place, they are active. So they are called the memory cells. Now we are coming to the activation of T cells. Okay, activation of T cells. Now this is the lymphocyte. Okay, it is a T lymphocyte because the lymphocyte is matured in the thymus gland, but now it is present in the blood. Now in the T lymphocyte or T cells, there are two types. Based on the function, we are classifying them into two types. Helper T cells, one type. Killer T cells, two types. Helper and killer. Now first we will take helper T cells. What they do? Once they are multiplied, they are releasing one chemical called a cytokine. Okay, that is, they are releasing one chemical called what? Cytokine. Now the cytokine activates B cells. Actually they are coming under what? T cells. But the chemical produced by the T cell is activating the B cells. Immediately the B cells are producing suitable antibody. So one T cell is always inducing B cells. B cells are always producing antibody. That is one type. Now we are going to see the killer T cell. Now in the name itself we can understand. The T cell is killing the pathogen. The killer T cell move over the body. That means what? It is circulating along with the blood throughout the body. Move over the body. Destroy the infected cells. Okay, what is the destroyed? Now directly this killer cells are going, the T cells are going, touching the microorganism and it is engulfing or damaging the cell, killing the cell. So destroy the infected cell or damage the cells, damage the infected cells. Now sometime what will happen? The microorganism will enter into our body cell. Already our body cells, some of the cells are destroyed, but microorganism is present. Even these killer cells are destroying our own body cells which are infected with microorganism, damaged cells. That is the role of helper cells and killer cells. Note, neutrophils and monocytes. Now neutrophils belong to granulocytes, monocytes belong to agranulocytes. The two type of white blood cells do phagocytosis. What is the meaning of phagocytosis? Engulfing and digesting the microorganism, but they cannot produce antibody. They cannot produce antibody. Now we are coming to the details of antigen and antibody. Antibody is a chemical produced by our immune system. It is our own chemical, our own body chemical. But antigen, it is a foreign chemical or foreign substance or foreign structure, anything you can say. Antigen, abbreviation AG. Now in the AG given is antigen, AG. Now that is the antigen. Antigen is any foreign substance I told you, coming from, which is not connected with our body, coming from outside. Now complex or large foreign molecules. The molecule may be complex molecule or it may be larger molecule, macromolecule, what we can say. Large foreign molecules that induce our immune system. Once they enter into our body, they are inducing our immune system, stimulating our immune system. First, that is a meaning for this antigen. Many meanings are there. The second point, these antigens are the molecules that can react with the antibody. Once the antibody is produced, antigen will always react with the antibody. Antigen molecule is a foreign molecule, antibody molecule is our body molecules. Both will react at the time. Which is more powerful, we have to see. If our antibody is powerful, the antigen will be destroyed. We will not get disease. Sometimes our antibody may be weak. The pathogen, the foreign molecule, antigen molecule may be powerful. At the time we are getting sick, we are getting disease. Now that is a molecule produced the immune system. Third point, each antigen is specific to antibody. This already we have studied. One antigen is there means one specific antibody will be there. Only they can react. They will never react with others. It is also called immunogen. Now see that term. The antigen is also called immunogen. Why we are calling it as immunogen? It is stimulating our immune system. That is called immunogen. What is the meaning of immunogen? Very shortly it is given. Factors inducing our immune system. Instead of telling the wrong one, factors inducing our immune system, we can tell only one common term, immunogen. So antigens are also called immunogens. Now antigen related terminologies, the foreign molecules, larger molecules. Now connected with that, some terminologies we have to study. The first terminology is haptens. Haptens. What are haptens? We have to see. Haptens are the substances, chemicals. Haptens are some chemical substances that are non-immunogenic. That means they are not stimulating our immune system. They are not stimulating our immune system, but can react with antibody. What is the meaning of it? It is not inducing immune system, but can react with antibody. Say that is the haptens of the chemicals, foreign substances. They are entering into the body. They can react with already present antibody, which is already secreted for some other purpose. With that antibody they can do, but it will never stimulate our immune system to produce new antibody. So they can react with the already existing antibody. Next one is epitope. Now what is the meaning of epitope? We have to see. Say antigen is a macromolecule we have studied. In chemistry you are studying molecule. Many atoms join together form one molecule. Now that is one antigen molecule is there. In the antigen molecule there are certain active part. Some active part means some regions are there. They are very, very active in doing their functions. So epitope is the active part of antigen, active region of the antigen. It is also called antigenic determinant. What is that antigenic determinant? It is the place which is determining the character of antigen, the property of antigen. That is called antigenic determinant. Based on this particular region, what is that epitope only? The type of disease will be caused in our body. Now we are coming to the third one, paratope. Now once the antigen is present, our body is producing antibody. Okay, in our body antibody is present. In the antibody also some active regions are there. Very, very active regions are there. So each active region is called paratope. So it is the active part of the antibody. In the antibody molecule, it is the active part. It is the antigen binding site. Actually, there is a one, what is that? Paratope is there. It is a very, very active site. The antigen's active site will come and join. They will react with each other. That only what is given here. It is the antigen binding site. Antigen is coming and attaching here. That is, it can recognize and bind with the epitope. First it will recognize whether the antigen is suitable for it. Recognize and bind with the epitope of the antigen. Both will bind. Once the binding is there, reaction will take place. If antibody is powerful, paratope is powerful, microorganism will be destroyed. If the epitope is powerful, microorganism is powerful, the paratope will be destroyed. That is, antibody cannot do any work like that. So these are the three terms we have to know what is the meaning of that. Haptens, epitope and paratope. Now we are going to study about types of antigen. Okay, lot of foreign chemicals, foreign molecules are coming, entering into our body, causing problem. They are called antigens. What are the various types? You have to say. First one, there are two types of antigens. The first one is exogenous antigen. Exogenous means these antigens are coming from outside into the body. These are coming from outside, from somewhere they are coming and they are entering into our body. Once they enter into our body, they are causing some problem. That is called exogenous antigen. Next one is endogenous antigen. There are certain antigens secreted by our body. Okay, antibodies are already produced, so that is different, that is for immunity. But apart from that, some antigens, special antigens are produced in our body, but they are not causing any problem, but they are helping us in some way. The antigen, if any antigen is produced inside our body, it is called the endogenous antigen. Now we will take first one, exogenous antigen. What are the antigens, foreign substances that are entering? Exogenous antigens enter into our body from outside. Example, what are they? Pathogens, microorganisms, toxins, toxins means poisons, venom. Now some poisonous animals are there. The poisonous animal like snake, scorpion, so many other animals are there. When they are biting, the poison is coming to our blood. That poison is called a venom. Pollen grains of the plants, some pollen grains of some plants we can call, that is an antigen. Pollutants, poisonous chemicals present in air, water and soil. They are called pollutants, etc. Now for example, water is contaminated. Lot of poisonous chemicals are there. We don't know about it, but we are drinking. It is a chemical, foreign chemical entering into our body. So these are all the exogenous antigens. Endogenous antigens are formed within our body. I already told you, this is not for immunity purpose. They are not for immunity purpose, but they are produced in our body. Endogenous antigens are formed within our body. Naturally, they are present. What are the example? Blood group antigen. When we study in genetics about human blood group, 4 type of blood groups you might have studied. A group, B group, AB and O. For that, which determine the blood group A or B or AB or O, the type of antigen for blood group. It is not antigen for poisonous or microorganism or anything. It is a chemical which is determining our blood group. It is natural, not causing any problem. Blood group antigens. That is endogenous.